Bismillahirrahmanirrahim. Assalamu alaikum. My name is Dr. Kuratulan, and this presentation is prepared and presented by myself, Dr. Kuratulan. My designations are as follows: as Research and Development Head, Regulatory Officer in DAP, and Business Development Head as well. Today, our topic of discussion is about drug discovery related to reduction use of drug and formulation strategy. First of all, we want to explain that struggles to explore new formulation, why there is need to develop or to explore new formulation. Pharmaceutical firms adopted diverse strategies to address these formulation challenges. The main goal remains to increase the chance of clinical success without increasing the time to market of the new drugs. Hence, they overcome different challenges side by side, thus formulate the best formulation. Next, I want to explain about rational use of medicines. Rational use of medicines. Rational use of medicine requires that patients receive medications appropriate to their clinical needs and doses that meet their own individual requirements for an adequate period of time and at lowest cost of them and their community. There are 6 R's in medicine use. These are as follows: as identifying the right patient, verify the right medication, verify the indication for use, calculate the right dose, make sure it's the right time, checks the right route. Rational use of antibiotics. All antibiotic use, whether appropriate or not, can promote the emergence of resistance in bacteria. Unfortunately, inappropriate and excessive use of antibiotics is common in both high and low income countries and in both the human and animal sectors. Objectives of research. Today, my research has certain objectives, and these objectives are as follows: as they are basically dependent upon introduction of some strategic guidance to a rational formulation strategy of new molecules and as oral dosage forms, based on scientific principles and factors determining the oral bioavailability. In addition, the industrial constraints are considered as increasing the productivity of pharmaceutical research is one of the major current challenges of the industry. Rational drug formulation strategy. A rational formulation strategy can reduce the developmental time cycle and increase the chance of success moving a new drug through a different preclinical and clinical phases. This approach has the advantage of streamlining the formulation process in order to avoid delaying the development of new drug due to formulation related issues. As we know that there are certain formulation related issues always come in our making a formulation. Thus, there is a need to overcome these all hurdles, and the strategy is thus made, which depends upon the certain parameters, or you can say there are certain preclinical and clinical trials should be done to overcome the different hurdles in between our rational formulation strategy. Rational approach development new drug. A rational approach to require tests as early stage may include there are certain tests are required which prove your formulation is authentic one. These testings are melting point, microscopy, solubility, pH 1, pH 2 7.4, log P, pKa, and permeability and chemical stability as well. What does do you mean by what does this is permeability contribution? What do you mean by this? As air solubility and permeability are major constraints to bioavailability. Bioavailability means the medications or medicine available in your drug or ready to absorb in your blood or body to perform its action. This is called the bioavailability. These both factors, solubility and permeability, controls your bioavailability. The permeability problems could be overcome by using different permeability enhancers, but their toxicity is often a limiting factor. If the effective permeability is very low, less than 5% of metoprolol FDA high permeability reference, the barrier for GI absorption is too high for oral delivery. Hence, to overcome all of these problems, thus an alternative route of administration may be considered. Thus, you can see that to overcome the permeability contribution factor or to overcome the permeability hurdle, we should either choose an alternative route to administer a drug or we should improve the permeability of a drug. because our GI absorption matters a lot in the availability bioavailability of a drug. What is dose solubility and how it's effective? As follow as we can suggest that permeability and solubility are the main both are the main authentic factors. Solubility also plays its vital role. The concept of dose solubility number is introduced as a tool for chemists to assess the developability of different molecules very early during discovery stage. Based on this understanding, a rational formulation strategy for preclinical and early clinical phase is provided. Solubility contribution. As we can say that solubility contributes as well, we can see some molecules with poor aqueous solubility and still have high bioavailability. This could be explained by the fact that the gastrointestinal medium is a dynamic one where the physiological factors such as enzymes and the compensating points of high permeability can be enough to correct the poor solubility. Hence, permeability and solubility run side by side in developing a new formulation. We manage the solubility by enhancing the permeability, or we can manage the permeability by enhancing the solubility. Our purpose is to increase the bioavailability of a drug in our body or in our bloodstream to perform its action. Evaluate the influence of solubility in the bioavailability of a compound. Is the absolute bioavailability of the compound in a simple suspension high? There is a strategy. In this strategy, in a suspension, is the absolute bioavailability of the compound in simple suspension high? We can see that the bioavailability is not solubility or dissolution limited, yes, or is the relative bioavailability in a simple suspension comparable to the bioavailability in a solution? No. So these are the different situations and we can handle them as follows as in first situation the bioavailability could not be further increased by enabling formulation. Hence in the first case the bioavailability is not solubility or dissolution limited because the drug is freely available. There is no need or no any type of further. Thus, the bioavailability could not be further increased by enabling formulation. But in second strategy or in second case, where the relative bioavailability in a simple suspension comparable to the bioavailability in a solution, thus there are three different situations by which we can increase the bioavailability of a drug component. Bioavailability solution increases Bioavailability solution is more than the bioavailability suspension. The bioavailability is or solubility dissolution limited. Bioavailability solution is equivalent to the bioavailability of a suspension. Thus, the bioavailability is not solubility or dissolution limited. In third scenario, bioavailability solution is lesser than the bioavailability of a suspension. The bioavailability is not solubility or dissolution limited. These are the three different strategies in which three different scenarios can be seen. In first scenario, the bioavailability could be increased by enabling formulation, and thus when we enable a formulation or when we making a formulation, this situation can be handled easily, and the bioavailability or solubility dissolution can be enhanced. This is the case when bioavailability in a solution is more than that the bioavailability in a suspension. In second scenario, when the bioavailability in a solution of a drug is equivalent to bioavailability of a drug in a suspension, then the bioavailability is not solubility or dissolution limited. Okay? And thus the low bioavailability may be related to permeability or first pass issue. Okay, we can consider in this case the low bioavailability may be related to permeability of first pass issue. In third scenario, bioavailability of a in solution is lesser than bioavailability in suspension. The bioavailability is not solubility or dissolution limited. The low bioavailability may be related to stability issues in GI tract. Why micronized compound required to make suspension? As we know that to make a suspension, a micron size of particle, 5 micron meter size of a particle matters. Why we need micronized compound to make suspension? If improving the solubility is necessary, evaluating the possibility of a salt formulation is the first recommended approach. Okay, you always minded that the salt formulation or salt formation in evaluating the possibility is first recommended approach. Okay, then the use of micronized compound is highly recommended too. Wherever a suspension is prepared, this is to improve the dissolution rate and helps obtaining a homogeneous suspension when the complete solubility is not obtained. Okay. In the case of suspension, when the solubility is not obtained, we would choose two different scenarios. These different scenarios are as follows here. To increase the dissolution rate, we recommend to use the micronized compound or micronized APIs to make a suspension. Decision-making brain development of any clinical formulation. As you know, that the new compound formulation is a very challenging task in our pharmaceutical firms or in different organizations or foundations. Hence, these are the multiple dimensions, or you can say these are the multiple ways by which we can evaluate our new compound. These different situations are either we can make salt, either we can make micronized over formulation, or surfactant lipid based formulation, co-solvent nanoparticle solid dispersion, or co-solvent lipid based formulation, or you can say nanoparticles or solid dispersion. Okay, now there are different types of scenarios are evolved, or you can say depict in this picture. Either there are in the surfactant lipid-based formulation, in which the log P is enhanced and the melting point is decreased. In co-solvent nanoparticle solid dispersion, when log P is decreased and their melting point is increased. In co-solvent lipid-based formulation, nanoparticles or solid dispersion, when there is increase in log P value and as well as increase in melting point. Thus, we can see that these all three last mentioned strategies could be handled by log P or melting point. No. What we can do micronization improves their solubility. There is a question at that point that either micronization improves their solubility to we can say that micronization of salt formulation, both formulation can be made by adopting these strategy if we micronization improve their solubility. Okay, but when the molecule is ionizable, no, the micronization improve doesn't then improve their solubility. So what we can do now? There is a salt screening improve their solubility. uh, yes, in salt formulation and in salt formation, salt screening improves their solubility, or the molecule is in ionizable form. Then you can make and salt formation. The molecule is ionizable. Bioavailability is solubility limited. Okay, yes, we can see that the bioavailability is solubility limit. There's a different physicochemical testing or characterizations can be performed on them to improve their bioavailability. In aqueous solution suspension, bioavailability of a drug is solubility limited. This doesn't matter in aqueous solution or in suspension form. Thus, by adopting these all steps, or we can move step by step, at the last we can approach or new compound entity or a new formulation. Conclusion: a rational formulation strategy can reduce the developmental time cycle and increase the chance of success moving a new drug through the different preclinical and clinical phases. This approach has the advantage of streamlining the formulation process in order to avoid delaying the development of new drug due to formulation related issues. In addition, it makes into consideration the practical constraints of the pharmaceutical industry related to the need of reducing the time to market while keeping the cost of development under control. Hopefully, you all got my point very well. Thanks for watching, and kindly subscribe and stay tuned with my channel. That is basically Research is Life.